So in this video, I'd like to talk about two important technologies by which we can silence a gene. So we sort of talked with GMOs, but GMOs are often a way of adding a gene to an organism, but here we're going to talk about subtracting a gene. So taking basically a gene that is already present in an organism, and then either totally deleting it or altering its expression. So one way to do that would be to study the gene. So if we, for instance, knock out activity of the gene, we can basically get rid of the protein the gene encodes and ask, how does that change things? Or ask, how does that change the gene expression of genes that are related? Or we can permanently modify the organism. So for instance, we might have a gene in cancers that is causing tumor growth, and so we could try to knock out that gene. One possible target would be there's a viral receptor that makes certain people susceptible to HIV and others immune to it. So one possibility would be to knock out the gene that makes people susceptible to it. Or for instance, in crops, we could try to knock out some gene that causes those plants to not grow as quickly. So here we're going to study two main ways. So the main temporary way we can do this is with RNAi. So this does not change the DNA, and it silences gene at the level of expression, whereas CRISPR changes the DNA, actually causes a mutational change in the DNA. So let's go through them one at a time. So RNA interference was discovered in the late 90s by Mello, Andrew Farr, and others, initially in the nematode C. elegans, for which they shared the Nobel Prize in 2006. And so this is a system where double-stranded RNA, okay, so remember double-stranded RNA is kind of weird. We're not accustomed to that. But where double-stranded RNA causes degradation of related RNAs in the nucleus. So we'll talk about that in a second. And this is thought to be an ancient mechanism to suppress double-stranded RNAs, such as viruses and transposable elements. So it also seems like it functions in translational control, and it's conserved across many or most eukaryotes. So here's the whole process. Let's take it step by step. Okay, so the first is to identify identify and cut up the double-stranded RNA. So imagine that we have a double-stranded RNA molecule. So in the nucleus somehow there's a double-stranded molecule. So this protein called dicer is actually going to cleave this into small double-stranded RNA. So that's the first step. And so we can pause here and think about why double-stranded RNAs. Well, as we know, double-stranded RNAs are not common in cellular gene expression. We don't expect to see them much in the normal functioning of a cell. But they do actually show up in viruses. Some viruses are double-stranded RNAs. And some transposable elements, which are these elements that replicate themselves through the genome and may cause problems, these elements can have stages that are double-stranded RNAs. So the idea here is that this is a process to identify these enemies, these invaders, and try to silence them. Okay, so dicer is going to then identify them and chop them up into small chunks. Okay, so each of these small chunks is going to get loaded onto this protein, and this is going to be the protein that's responsible for identifying other copies of these molecules and getting rid of them. So basically what happens is if you find a double-stranded RNA, then this complex, which is called RISC, is going to identify all copies, double-stranded or single-stranded, of this RNA and degrade them. Okay, so yes, the small interfering RNAs assemble into ribonucleoprotein particles. So that just means RNA protein complexes. And these are called RISCs for RNA-induced silencing complex. And initially, the RISC complex will not be active, but that's our next step. So our next step is that one of these strands gets stripped away, just leaving the RISC with this one short single-stranded RNA. So this complex contains argonaute proteins. You don't need to worry about that, but you will probably hear about argonaute proteins in your future classes. And so we call these ssRNAs, these short single-stranded RNAs. Okay, and so now we've got this thing that has this exposed 22 to 28 nucleotide sequence, and it's actually going to go, because the sequence is complementary, or it has a sequence it would be complementary to, it's going to find RNAs that it's complementary to. So it's going to identify RNA molecules that it's complementary to. And when it identifies them, it's going to bind to them. So here it is bound to them. And then once it's bound, now the RISC is going to basically sabotage this RNA. And so in different species, it works differently. It seems like for different binding pairs, it works differently. But one thing it can do is actually chop up the mRNA, cleave it. And the other thing it can do is it can arrest translation. So it'll stop this mRNA from being translated. So in either case, what's happened is we identify a double-stranded RNA. This RISC complex, or the whole RNAi machinery, says, hold on, this is surprising, this doesn't seem right. And so we're going to then become suspicious of this sequence and then go around the cell and silence other RNAs that contain the sequence. So that's basically, that is the flow of RNAi. So this is very important. When it was discovered, this is a brand new form of translational regulation. Explains a lot of how transposable elements are controlled. And it allows, again, transposable elements are these elements that copy themselves through the genome. It allows for experimental knockdown of virtually any gene in basically any organism where you know what gene you want to knock down, where you know the sequence. Because you can just feed these organisms. In the case of worms, you can just feed them double-stranded RNA, and they will shut off the gene. So this opens research to study of non-model organisms, and now we're no longer confined to obtaining random mutants. We can target a gene. And potentially, we could have therapies that use RNAi to turn off genes whose misexpression, whose incorrect expression, is causing medical problems. Okay, so that's RNAi. So now let's turn to CRISPR. So CRISPR is another DNA cutter. So we talked about restriction enzymes and that cut DNA, and CRISPR similarly is going to cut DNA. But unlike restriction enzymes, CRISPR can be targeted, as we'll discuss. So restriction enzyme, a given enzyme will only cut at a certain sequence, but CRISPR can be engineered to cut any sequence we want, basically. So CRISPR, CRISPR is a technology that we now use, but we stole it from an organism. We stole it from bacteria. So it's sort of part of the bacterial immune response against viruses. It's a way that bacteria identifies and chops up viruses so they can't burden the cell. And so it has two essential components, as we'll discuss. It has an RNA that's complementary to the target DNA, and then it has a protein that actually does the cleavage of the target DNA. Okay, so this is the best figure I could find for now, so let's go through it. So what we have here, so in blue or aqua, that's the protein. That's the Cas protein, Cas9 as it's called. That's actually going to do the cutting. And then the second bit we have in purple here is what we call the guide RNA. So this is the RNA that is going to, you can notice here that this RNA is complementary to one strand of the DNA. So this is the DNA that's going to be targeted. And it's found due to complementarity between this region of the DNA and this chunk of RNA. And so when Cas9 and its associated guide RNA find a targeted DNA sequence by complementarity, it will then cleave the DNA. It will cleave the DNA, and then so we can do various things here. But the main point is it will cleave the DNA. So the simplest thing we could do would be to just knock out this gene. So it will cleave it, and then it'll get stuck back together by the cellular machinery, but probably with some errors. And so this allows us to do targeted genome editing on all sorts of organisms, from bacteria all the way to humans. And so in conclusion, our recurring theme here is that biotechnology tends to borrow genes or proteins that were initially evolved in the arms race between microbes and viruses. So we didn't talk about, but penicillin, you may know, is a substance that was evolved by fungi to kill bacteria. We talked about restriction enzymes, which were evolved by some bacteria to kill other bacteria. We talked about vectors, which was the plasmid that was involved in gall formation. So vectors were used by viruses in their war on plants. And then RNAi, which is eukaryotes evolved to escape viruses and other elements. And then we talked about CRISPR, which bacteria use to destroy viruses. And so here's our summary. We can just compare and contrast. So RNAi is temporary, whereas CRISPR is a permanent mutation. RNA involves translational regulation, whereas CRISPR involves causing mutations in the target DNA. CRISPR targets RNA, RNAi targets RNA, CRISPR targets DNA. But then they have these two. They also have similarities. So both use RNA complementarity to find their target. And then finally, RNAi was initially discovered in eukaryotes, whereas CRISPR is mostly confined to prokaryotes.